This is Sexual Reproduction and Meiosis, Chapter 11. What we're going to find in this chapter is that many of the processes we'll be discussing we've already covered in mitosis. The steps are similar, there are just variations within them. When we talked about mitosis, we talked about cells that were being created for growth, for development, and for repair. In meiosis, we are creating cells that will be used specifically for reproduction. We're going to produce gametes. This is egg and sperm. Gametes are different from the other cells of the body because they are haploid, meaning they have only one set of chromosomes. Mom's chromosomes if it's egg, dad's chromosome if it's sperm. When the two gametes join during fertilization, they will become a diploid cell. That diploid cell is also known as a zygote. Keep in mind that because they are diploid, we now have homologous pairs of chromosomes. The process of meiosis is very similar, as I said. We'll begin with an interphase period, a G1, an S, and a G2 phase. Everything that happens in mitosis during that period also happens in meiosis. After interphase, that's when the cells actually enter meiosis. They'll begin at meiosis 1. Each of those two rounds, meiosis 1 and meiosis 2, will have the same type of division spots: prophase, metaphase, anaphase, and telophase. Our first step is prophase 1. Prophase 1 is very important because this is where the stage is set for what the new organism will look like. It's similar in mitosis at the beginning because the chromatin will thicken and become chromosomes, the nuclear envelope or the nuclear membrane will disappear, and spindle fibers will form. After that is where it becomes different. We'll have a thing called synapsis and a process called genetic recombination. In synapsis, we have chromosome pairs being matched up, meaning chromosome number 1 of mom, the same chromosome of dad. How this process is done is still not fully understood, even though cellular biologists have been studying this process for many, many years. Once the chromosomes are found each other, they will stack up. It doesn't matter whether the maternal or the paternal, mother or father chromosome is on the top of the stack. It's a random situation. Once they're stacked up, proteins will form in between them. These cohesion proteins will glue the two chromosomes together. That glue, that protein in the center, is called the synaptonomal complex, and it'll be used at the most portion of meiosis to move the chromosomes together as a pair. Genetic recombination is also called crossing over. We talked about this a very, very short amount of time when we talked about DNA replication. In this case, the chromosomes, mom and dad's number 1 and mom and dad's number 12, like I said, are already stacked up because of the synaptonomal complex. And the chromosomes will cross over, exchange information, in a sense like twisting up, and then genes from one chromosome will exchange places with a similar gene on the other one. This happens on non-sister chromatid, meaning mom's information is exchanged with dad's information. That crossing over site is called the chiasmata. There can be one crossing over site as in the picture, or there can be multiple sites. It depends. Again, this is a random thing. This is really important because then you have a chromosome that's a combination of mom's information and dad's information. That gives the organism a much greater variety of genetic possibilities. The chiasmata will stay attached until anaphase 1. Our next step is metaphase 1. This is very similar to mitosis. The spindle fibers are going to stretch out and they're going to attach to the kinetochores. What's different here is that they attach on the same side, one for each homolog, not one for each chromatid. This allows the entire chromosome to be moved back and forth without applying any pressure to the centromere. The fibers are going to move the homologous chromosome pairs back and forth, a little bit toward the center, a little bit back, just like they did in mitosis, until they are lined up along the metaphase plate. How those chromosomes are lined up, meaning is it all the maternal on one side, all the paternal on the other, or whatever variation randomness can have, is going to make up the new organism. If you look at this picture, imagine a line going right between the center of those two chromosomes, the metaphase plate, and you can see the variety of different possibilities of the new organism. In anaphase 1, things are going to begin to break down. The cohesion proteins, the synaptonomal complex, is going to break down, which is going to allow the chromosomes to begin to move. The chiasmata are going to break down, and so the two pairs are now separated. The microtubules are going to begin to shorten, and the entire chromosomes are going to be moved to opposite poles. Sister chromatid remain attached, so we're moving entire chromosomes, the original and the chromatid, to the poles. When an anaphase 1 is complete, you'll have one full set of chromosomes at each pole, and each chromosome still looks like an X, meaning that the sister chromatid are still attached. Because of the randomness of where the chromosomes were matched up, remember the previous slide, what you end up with here at the end is completely random. A cell can have all of mom's chromosomes, all of dad, or any possible combination. In telophase 1, we get prophase 1 back again. The nuclear membrane reappears, the nucleolus reappears. The sister chromatid are cram-packed at each pole, but keep in mind they are no longer identical because we've had that crossing over, the genetic recombination. Cytokinesis may or may not occur depending on the species. For humans, we do have cytokinesis, so the cytoplasm, the organelles, are all divided up, and we end up with two haploid daughter cells. There'll be a resting phase, and then the cell will enter meiosis 2. That resting phase is species dependent. This picture is just a summary of what I just talked about. It has all the different phases, and then it has in detail exactly what happens. You can see this in your book if you want to have a refresher. This picture is actually a really good explanation of what we just talked about. Meiosis 2, same thing as mitosis. We go through each of the stages, prophase, metaphase, anaphase, and telophase, and everything is exactly the same. In this case, in anaphase 2, the sister chromatid are separated so that you have the individual chromosomes and their duplicates separated. When meiosis 2 is over, telophase 2 is over, you have 4 sets of daughter cells, each with their own set of chromosomes. After telophase 2, then we'll actually go through cytokinesis, and we'll have 4 individual haploid cells. And this again is a picture, just kind of a short version of what we see. We start out with a normal cell that has both mom and dad. We go through S phase of interphase, and those cells are duplicating the DNA, so you have sister chromatid. After meiosis 1, we've split those chromosomes, so that we have an individual set of chromosomes in each cell. This is called reduction division because we've gone from mom's 1 and dad's 1 to just one or the other. We go through meiosis 2, we separate sister chromatid, so we have one original and one duplicate. This is not reduction division because we're not reducing the number, we're just separating the original from its copy. Remember I told you that the AP people love comparisons. Well, they love to do a comparison between meiosis and mitosis. There are 5 that we're going to talk about. First, in meiosis, homologs are paired, synapsis, during prophase 1. There is no pairing of the homologs in mitosis. Number 2, there is crossing over or genetic recombination in meiosis. Doesn't happen in mitosis. Number 3, the kinetochores are attached at the spindle fibers on one side rather than 2 sides, so that the entire chromosome is pulled to the pole. Number 4, sister chromatid remain joined at the centromeres all the way through meiosis 1. And number 5, there is a second interphase between meiosis 1 and meiosis 2, and there's only one interphase in mitosis. That means that there's only one replication of DNA. This slide and this slide, these are just a visual representation of the difference between meiosis and mitosis. Gives you another way to look at it. The last little bit we'll cover is a mistake. Mistakes happen. Non-disjunction means that instead of mom's chromosome going to one cell and dad's going to another, they both go to the same cell. That means you end up with an extra chromosome in one cell and not enough in the other. Anytime you have a gamete that has a missing or an extra chromosome, those are called aneuploid gametes. A lot of times this is what causes miscarriage because there's just too big of a mistake for the cell to make and to be able to handle. Those that allow organisms to continue to grow create many of the disorders that we know about, such as Down syndrome, and we will talk about Down syndrome more specifically in class. The last thing I want to cover is asexual reproduction. That's the production of identical copies of the parent. You have a thing called parthenogenesis and cloning. Parthenogenesis is where the female will lay an egg that hatches into a new baby without there ever being any fertilization. The offspring are genetically identical to the female, and this usually happens when there are no males present. For example, in honeybees. Honeybees have 2 types of offspring, female workers, which come from fertilized eggs, and male drones, which come from unfertilized eggs. It takes 2 X's to make a female, so if the honeybee lays eggs without being fertilized, they only have 1 X. That makes males. Komodo dragons do the same. If there are no males, they'll lay eggs. The offspring are genetically identical to the female, but again, they are male because you have to have 2 Z chromosomes to be male. And a female who lays an unfertilized egg lays male eggs that have only 1 Z. In cloning, you have a thing called budding. And budding means that you grow a little bitty, teeny tiny exact copy of yourself out of your side, and when it's fully formed, it breaks off and becomes a new individual. Jellyfish will do that. Then there's fragmentation, where a part breaks off the parent, and that part grows into a brand new organism. Tapeworms do that. Starfish do that. We also have artificial cloning that we do in the laboratory, and again, we will talk about that in class. That completes Chapter 11, Meiosis.